In this video, we will discuss about the reason behind elevated OXPHOS in the acidity to knock down pancreatic tumor cells. And from the heading, it is clear that it's the fatty acid oxidation that cause elevated OXPHOS in the acidity to knock down pancreatic tumor cells. In the previous video, we have understand that in the case of acidity to knock down cells, there is a elevated OXPHOS has been observed. So in order to check the reasons behind this elevated OXPHOS, they target three different pathways that are uh, that are available in the literatures that will cause elevated OXPHOS. That is fatty acid intake and glucose intake and the glutamine intake. So they inhibit each of these pathways individually and also in the combined to find out whether it will cause any effect on this elevated OXPHOS in the acidity to knock down pancreatic tumor cells or not. In order to inhibit this fatty acid pathways, they use etomoxib inhibitors, and to inhibit this pyruvate pathways, they use UK5099, and to inhibit this glutaminase, they use BPT-ES. And what they have find out from the oxygen consumption rate analysis of the muscles is that in the SETD2 knockdown cells, the etoximer has caused a significant reduction in the elevated OXPHOS compared to the other inhibitors. And these things they have also validated when they check the effect of inhibitors on the tumor suppressions. All the inhibitors shows some degree of tumor suppressions, but the etoximer shows a highest degree of tumor suppressions, and these things are quantified in terms of this tumor weight on the right hand side of the panel B, where we can see that when in the case of etoximer, the tumor weight reduction is highest compared to the other inhibitors. And thereafter, once they identified that is the fatty acid oxidations could be the reasons behind elevated OXPHOS, they wanted to check whether there is a lipid-rich environment in the SETD2 knockdown compared to the SETD2 wild type is there in the tumor microenvironment or in the stroma of the pancreatic tumor cells. So when they check this number of adipocyte-like cells in the SETD2 knockdown and SETD2 wild type, they find out that the number of adipocyte-like cells are higher in the case of SETD2 knockdown compared to the SETD2 wild type, indicates that there is a lipid-rich environment could be seems to be higher in the case of SETD2 knockdown pancreatic tumor cells compared to the SETD2 wild type cells. And when they check and these are the H&E staining, and when they check the immunostaining using the markers for the cancer-associated fibroblasts such as PDPN, which are found in the stroma of the pancreatic tumor cells, and the CK19 is the marker for the pancreatic tumor cells, and DAPI is for the control, it's the marker for the nucleus, and these PLIN1 and PLIN2 are the markers for the adipocyte-like cells. And what they have find out in the case of PLIN1, that the cancer-associated fibroblast and PLIN1 markers are coexist in most of the cases, and indicates that the adipocyte-like cells and cancer-associated fibroblast are exist in the stroma of the cells and not in the pancreatic tumors, or not inside the not in the pancreatic tumor cells. And for the PLN2, they do not see complete overlap of this PDPN and PLN2, but they also do not see any overlap of this PLIN2 with this CK19, indicating that this PLN2 markers positive cells are also in the stroma of the cells, not in the pancreatic tumors. So, and these things are quantified, and the quantification of this PLN1 and PLN2 in the SETD2 knockdown and SETD2 wild type cells is shown on the bottom right corner of the panel D, where we can see that in the case of knockdown there is an elevated level of PLIN1 and PLIN2, and together this panel D figure indicates that the more lipid like rich environment are formed. More lipid rich environment are formed in the stroma of the pancreatic tumor cells or in the tumor microenvironment, but not inside the tumors. And these things are observed in the SETD2 knockdown cells more compared to the SETD2 wild type. And thereafter, they check another markers for this lipid-rich environment, that is BO-DIPY markers, and they can see that in the case of SETD2 knockdown cells, they see the high BO-DIPY cells compared to the control case, that is SETD2 wild type pancreatic tumor cells in the and so this is also in the stroma of the cells, not in the not in the pancreatic tumor cells. And thereafter, after that, they check the other important other some characteristics of this lipid-rich environment, such as triglyceride, Ne, FAs, and cholesterol levels, and they find out all of them are higher in the case of SETD2 knockdown pancreatic tumor cells compared to the case of SETD2 wild type. So this indicates that lipid-rich environment are formed in the SETD2 knockdown pancreatic tumor cells, and that will cause the fatty that will enhance this fatty acid oxidations that leads to the elevated OXPHOS in the pancreatic tumor cells. So I hope you understood how they validate this statement, that is fatty acid oxidation cause elevated OXPHOS in SETD2 knockdown pancreatic tumor cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.